Hi, everybody. Sorry I wasn't able to be there in person today. I think that this would have been a fantastic workshop to attend at EMSL, but I'm glad to be able to join you remotely. I'm glad that this was an option. And I'm going to be talking today a little bit about some of my prior work in really high resolution imaging of organo-mineral associations in soils. And I think one of my goals for this is to provide a perspective from a potential user, sort of the broader scientific community, and give a little bit of my own perspective on scaling of more conceptual models and hypothesis-driven work, particularly in the understanding of soil carbon cycling and soil ecosystem processes. And I'm hoping that you might be able to have some feedback for me on what we did for this process and better ways that it could be done, which I'm sure have developed since this work was done. So, to start, I often kind of explain what I did for my PhD work to other people, and they get progressively more and more confused looking when I kind of go through the step-by-step recipe, as it were, of what we needed to do to prepare soil aggregates for imaging, because it just is kind of absurd when you think about it. So taking a tiny piece of soil with a pin, putting it in water on a super special pedestal, nothing else will do, making a slushy with liquid nitrogen, plunging it in there, slicing it up with a diamond. My favorite part, plucking an eyelash, yes, our own eyelashes, and making a little eyelash brush, gluing it to a stick, transferring that, and then 1,000 other steps. And just kind of to give a perspective from an outside audience, sometimes it's really unclear why this kind of effort is really necessary. What kind of information do you get, and why do we do all of this to be able to look at really tiny, tiny, tiny little pieces of soil? So for this talk, when we're thinking about scaling and thinking about the relevance of really high resolution measurements for broader picture, bigger picture ecosystem understanding, I think it's fun to think about, why go through all of this? And I think from my perspective, the way I approach kind of the value of chemical imaging and microscale measurements is first thinking about it as a way to provide really direct evidence, direct visual evidence for processes that otherwise are mostly conceptual models or hypothesized processes. And then also, probably more importantly, to provide new ideas and new hypotheses that can be tested at larger spatial scales. And so for today, of course, because of this emphasis on scaling, I'm going to give some thoughts and some new projects that are in motion as a result of new information that was generated only through this process of really microscale imaging. And so for today, I'm going to give kind of a really in-depth example of just one project, and thinking specifically about how it could be used, or how these measurements were used to look at direct evidence of interaction mechanisms, and also new ideas and hypotheses that were generated from this work in this kind of dual benefit concept. And then also just added a little bit at the end on some new work and some more specific and more quantitative scaling efforts with some measurement model integration that we're working on through an EMSL and a DOE project. So to start, this first kind of in-depth case study I'm going to talk about is the result of some of my PhD work at Cornell University, working in soil and crop sciences with Johannes Lehmann and Akio Enders, a fantastic technician. And we developed all of this through a collaboration with the Cornell Applied and Engineering Physics program, the Center for Materials Research. And that was really essential for development of some of these approaches. And this is a published project that you can find if you're interested at this link. So to back up a little bit, the motivation behind this project was to understand some of the kind of core mechanisms, basic processes that lead to soil carbon stabilization and persistence. And that's important because soil is an enormous carbon reservoir. We often quote the amount of 2 to 3 times the amount of carbon in soils than the atmosphere and vegetation combined. But it's not a static pool. It's gaining or losing carbon based on the balance between inputs and losses. And one of the more kind of long-lasting pools of soil carbon is that is associated with reactive metals. And in some estimates, up to 70% of that carbon is associated with metal interactions. So this process of stabilization could be a potentially really important process that's conveying long-term persistence to carbon. And so these carbon reactive metal associations are our focus area for our imaging project. And so I'm going to start with kind of a cartoon image of what the sort of hypothesized model going into this project was. And so we know that reactive iron and aluminum minerals tend to form relatively stable associations with carbon because of the interactions between carbon functional groups and hydroxyl surfaces on short-range ordered iron and aluminum minerals. We also have some evidence that that is particularly facilitated by organic compounds rich in nitrogen. So we expect that there could be accumulation of more nitrogen-rich organic matter directly at that interface between iron and aluminum minerals and organic matter. That could be sourced potentially from microbial residues, essentially dead microbes. And another element of this hypothesized model is this idea of a layered kind of accumulation of organic matter beyond sort of just a simple surface coverage, but the accumulation of organic matter in layers, which would allow for sort of additive building of interacting carbon on these mineral surfaces. So this is kind of the model going into it. And I put that asterisk there just to kind of further underscore that the idea behind some of this chemical imaging is to help provide direct evidence for what we might have as a hypothesized model. And so for this work, we were looking at volcanic soils, which are particularly high in reactive metals and also tend to have large amounts of relatively old soil carbon. Andesols, volcanic soils in the US are pretty limited in their spatial extent. So they're just shown here in hot pink on the map. So a bit in the Pacific Northwest, of course, and a bit in Alaska and across the islands of Hawaii. But what's important to note is that even if it's a relatively small spatial extent, volcanic soils store a disproportionate amount of carbon. And also these reactive metal minerals are present in basically all soil types in different forms and in different abundances. So it's a relevant process beyond just these case study soils. So the goal of this study was to try to get to be able to directly visualize the interface between reactive iron and aluminum and carbon, being able to sort of verify these hypothesized mechanisms of nitrogen-rich organic matter facilitating the interaction right at the surface, and also potentially this additive accumulation in layers. So the problem with this for really high resolution imaging is that, of course, soil organic matter and soil matrices are incredibly spatially and chemically complex. And so there are a variety of different approaches that we could use to try to look at some of the architecture and spatial arrangement. So with EDX, electron dispersive X-ray spectroscopy, we can get great good spatial detail, but no ability to actually show the speciation of carbon, what kind of carbon compounds are present. With techniques such as X-ray absorption spectroscopy, we can get information, chemical information about carbon forms, but potentially are limited by spatial resolution, not being able to probe, the sub-nanometer type scale needed to look directly at the interface. So for this project, we used a combination of EDX for kind of more overview mapping, and then at the very high resolution scale, scanning transmission electron microscopy with electron energy loss spectroscopy, STEM EELS, which has an ideal really, really small sub-nanometer scale spatial resolution. In crystals and other sort of more regular materials, potentially down to angstrom scale, single angstrom or less. For soils, which are definitely not uniform, uniform crystal type materials, we were aiming for sort of 0.1 to 1 nanometer spatial resolution. And combined with this really high spatial resolution, we also have the ability to probe carbon composition, because EELS is a technique that measures inelastic scatter, which is related to the bonding environment of elements in the material. But this is a transmission approach, so we needed to get a super, super thin electron transparent imaging area. And so with soils, this is a huge challenge because of the inherent heterogeneity and carbon spatial distribution and chemical characteristics. We wanted to be able to maintain spatial information, so micropores, organic matter distribution is really important. We wanted to prevent damage from high energy techniques, particularly for these kind of soft elements like carbon. And then we're looking at carbon, so we also want to try to eliminate carbon-based resins or stabilizing materials. As I'm learning, there have been some developments in alternative resins, but our approach was to try to eliminate a carbon resin entirely by using an entirely cryogenic sample preparation process from initial thin sectioning all the way to STEM EELS imaging. And so the goal of this was then to stabilize the aggregate in water rather than an alternative material. So this kind of goes back to that, that recipe of step-by-step processes that I was talking about earlier. We started by embedding an aggregate in water and freezing it in slush nitrogen to prevent formation of ice crystals. And we started with a relatively coarse thin section to 1 to 5 micron thickness using a ultramicrotome diamond blade under cryogenic conditions, which was transferred to a copper TEM grid. And this is where the eyelash brush comes in. As it turns out, the rigidity and size of an eyelash is just perfect for transferring a tiny, tiny little curl of ice from a droplet to the TEM grid. And this is where our technician Akio Enders really had the fine touch in this process, which was really quite finicky. So then after the initial thinning process, we then transferred cryogenically to a cryogenic FIB instrument, which is using a gallium ion beam to further mill and thin the material. And we used 2 basic different geometries for FIB milling, generating a wedge, which basically results in a very thin area just at the edge of that, kind of angular section, or a lamella where we were thinning above and below just to generate a very thin bridge, essentially, of material that would be electron transparent for imaging. And so this, again, is a fairly time-intensive and finicky process, and maintaining cryogenic conditions from thinning into the FIB takes some specialized equipment. So this is where we really had to lean on collaborators in the material science program to help develop these techniques. And so then finally, we go from the cryo-FIB into a cryo-STEM EELS instrument. And this is where we can start to look at the overview image first, using EDX to kind of give us some context for where we want to look in finer detail. And so we have an overview elemental map that shows a region where there's a carbon concentration and an aluminum concentration adjacent to each other. And so we chose that area to then zoom in for a higher resolution electron imaging. We conducted a variety of different measurements, so point scans, line scans across the interface between an aluminum mineral shown here with sort of platy structure and a carbon region. And in that, we're able to look directly kind of at that interface between aluminum and carbon, which was an exciting methodological advancement, I think, for looking at these complex materials. So with those measurements in multiple locations in the same sample, and then across a couple additional aggregates that were prepared in similar ways, altogether, we were able to use that information to come up with some new insights into how organic matter might be arranged at these reactive metal surfaces. So coming back to kind of our prior model, we had a concept coming in of maybe a layered arrangement of carbon forms extending outward from the surface. We were expecting to see maybe nitrogen enrichment at the interface. And so in some ways, our observations kind of confirmed that, where we saw nitrogen enrichment, both at the interface between the mineral surface, shown here in gray, and the organic matter region. But we also saw something new, which was instead of these sort of linear structures, more kind of irregular, patchy soil organic matter structures that were interacting with other organic matter forms. And so we started calling these organo-organic interactions instead of just organo-mineral interactions. And what was interesting, though, is that we also found nitrogen enrichment at the interface between those phases. And so nitrogen at the mineral organic interface and nitrogen enrichment at these organo-organic interfaces. But what was interesting and what we weren't expecting was that the kind of accompanying organic matter composition was distinct between these two different kinds of interfaces. So more oxidized carboxylic acid-rich organic matter at the mineral interface, but more aliphatic, reduced organic matter forms at the soil organic matter patches. So what does this mean for our kind of coming back to these main goals of the value of high resolution imaging? So we were able to provide some direct evidence for these hypothesized processes and mechanisms. And also by looking at kind of the specifics of organic matter forms that are present at the interfaces, it provides some new ideas and hypotheses that we can test now in a broader scale. So I'm going to talk a little bit about the ongoing work that we're doing that's applying this information specific to new experimental design. And because we're thinking a lot about scaling here, I kind of sat back and thought about it a little bit more broadly. And I would, say that what I'm talking about here is more scaling of concepts and hypotheses rather than a more quantitative data, propagation type scaling approach. But I would argue, and I think that what we can call from these observations are scalable information. So we have scalable chemical information, what functional groups are located at the interface. And also scalable spatial information, what do these interfaces look like? Does it matter whether it's more sort of 2D accumulation in layers versus irregular blobs, which is what we found? And so I think that is really important information, if not necessarily a quantitative scaling type approach. So with respect to chemical information, I mentioned that we found kind of this distinctive enrichment of nitrogen-rich aliphatic carbon at the organo-organic interfaces versus nitrogen-rich carboxylic acid oxidized carbon at the mineral interface. And so this information was a new thing, and it led us to think about how molecular composition specific to nitrogen and these specific compounds would influence the formation of mineral-associated carbon, looking at gradients in organic matter inputs. I'm showing here across different potential tree species, or you could think about it in terms of gradients of agricultural crops and cover crop mixtures, which might have different nitrogen chemical profiles, organic nitrogen. And this project is now leading to the PhD work of a PhD student at Virginia Tech, Sophia Nikolakis. And she's mostly interested in how gradients in organic nitrogen chemistry in cover crops could impact the formation of mineral-associated organic carbon. And what we're looking at, though, more specifically than just kind of a broad nitrogen composition, is the specific, gradients in the composition of things like cytokinins, which are nitrogen rich, but a little bit less oxidized than a lot of amino acids, for example. And so what the microscale imaging work allowed us to do was kind of broaden our scope of potential target compounds and potential measurements of relevance for composition of nitrogen assessment. And then this project is also going to be using isotope-enabled techniques to track the fate of nitrogen from inputs into mineral-associated organic carbon. And so this work is just going forward now. And so stay tuned for more about how these measurements might kind of either align or not align between what we saw at the microscale. But I wanted to include this as an example of a really, I think, sort of direct link between something that was completely not known before, was enabled by microscale observations, and is now enabling us to test this at a larger scale. So to summarize this component, we saw that high resolution imaging allowed for evidence for the role of nitrogen containing compounds. We showed that rather than kind of a nice linear 2D arrangement in space was really patchy. And these both have implications for our conceptual understanding and design of new experiments. So to kind of take a little bit of a shift towards some newer work and a little bit more quantitative measurement model integration, I wanted to include this partially because Fiona Ellsworth is currently at EMSL. I don't know if she's actually there in the audience. Hi, Fiona. And conducting these measurements basically as we speak. So this is all very hot off the press and very much kind of ongoing work, but I think gives a good example of how users and the broader scientific community might benefit from generating measurements that could be really directly plugged into model assessment and model development. And so this project is led by Richard Marinos. He's the lead PI. He's at University of Buffalo, a variety of Buffalo collaborators. Jim Moran at Michigan State has been collaborating with us, and very many EMSL collaborators that I didn't want to list here. This is a little bit hot off the presses, and so I don't want to miss anybody, but many folks at EMSL are working with us to develop these measurements for this project. So the question is, it's a little bit of a shift from the organomineral associations now to think about root carbon and carbon compounds released by roots. And so the broad question here is, how does root carbon exudation respond to drought? And so there's an idea that with root redistribution of water under drought, which is termed hydraulic redistribution, this might alter the amount and also the composition of carbon compounds that are released from the roots as exudates. And so under hydraulic redistribution conditions, maybe we have water flux out of the root and a change to the diffusion and advection transfer of carbon compounds that are being released. And so this project is really a very cross-scale measurement and modeling project. On the finer scale, we're interested in generating measurements of how far carbon compounds move from the root, the extent of the rhizosphere with respect to carbon compounds, the composition of those exudates, sort of what's present, and then how they're distributed along the root. These are just, we're so early in the project, and so I'm not really even showing any data as of yet, but just to show that we're going to be using laser ablation, IRMS, to measure how far carbon moves using isotope labels and detection. And that's what we're working on with Jim Moran. And Fiona is there now at EMSL doing a lot of work with MALDI to look at the composition of those root exudates along the root. So both what is there, and then also how are they spatially distributed. The reason I wanted to show this, even though it's at a really early stage, is that we really thought in a lot of detail about how we were going to use these measurements in advance, before we actually collected the measurements, to inform model development at increasing spatial scales. So these measurements are being collected basically at the root soil interface. And then we're interested in scaling those up to the root and the plant scale, looking at distribution along the entire root network and in distribution in space. And then even taking that further to the landscape scale to look at how it might vary across landscape positions. And so this is an experiment and modeling approach that really tries to take specific microscale measurements and use them to enable model development. So I'll just give a couple examples about how we're doing this. Thanks for the note on timing. We're using a rhizosphere model corpse to look at carbon transfer in the rhizosphere, and a plant landscape model, parflow trees, to look at the landscape scale. And I just wanted to show, and this is a lot of detail, so don't worry too much about the specifics here, but what we were trying to think of is what is the knowledge gap at the small scale? How does the model currently represent this? What do we want to do to it? And then what kind of experimental data are we going to use? So as an example, the corpse rhizosphere model looks at a fixed radius. We want to make a variable radius around the root. And to do that, we're going to use MALDI imaging and LA-IRMS measurements. The current trees model is just exodic flux proportional to total root area, but we want to modify that based on root order. So again, MALDI imaging is going to play into that. And then finally, we're looking at the landscape scale, which is an entirely modeling exercise. And so this is just a really, really, really quick snapshot. I really wanted to include it mostly because Fiona is there at EMSL. I would love for folks to talk with Fiona and get some feedback on this project and what opportunities we might have for other integrated imaging possibilities. So please stay tuned for more. So with that, I'll wrap up, and thanks so much for having me there. And it's exciting to see as an external user capacities at EMSL and other locations. And I'm happy to discuss further. Thanks so much.